For this local expert presentation, I will be discussing direct RNA sequencing using Oxford Nanopore devices. Direct RNA sequencing involves a different way of sample prep that allows you to directly sequence RNA without the conversion to cDNA. Because of the nature of direct RNA sequencing, nanopores are the only method to use if you want that kind of data. So what is nanopore sequencing? The coolest thing about this type of technology is that it allows for real-time sequencing of DNA or RNA. No matter where you are, you can get your sample sequence without having to worry about proper lab procedures, having the dogs out, so you can wear your open-toed shoes, your Birks, your Crocs, and there is less technical training when operating these devices, so you can send undergrads out to sequence your stuff. So what happens here is I'll go over more of this in the next slide, but basically we have a helicase motor that you can kind of see here in this diagram to the right, and it powers the movement of RNA or DNA if that's what you want to sequence through a small protein hole called a nanopore. As this happens, an electrical current is converted into an RNA sequence by a super intelligent neural network. These ion currents, the electrical currents that produce the signature, can be read and interpreted in real time, so you can know if you've got killer anthrax or just regular suspicious white powder. Imagine if we had these in 2002. So let's look more at the specifics of this technology. I've already mentioned what nanopores are. They are embedded on an electroresistant membrane that is attached to the flow cell. And these nanopores have their own electrode that is connected to a chip that then measures the electrical current. When your DNA or RNA pass through the pore, it disrupts the current and produces a specific signature that corresponds to A, C, G, T, or U, depending on what you're using. Base calling algorithms are then used to determine the sequence of your sample in real time. Again, pretty cool. The GIFs to the right show a sample entering the nanopore. The nanopore is in the dark blue, and the sample is in the light blue, and then you have the adapter ligations in purple that help it bind to the sample to bind to the nanopore. And then as it's moving through the pore, you can see that the electrical current is being generated. You can see the different signatures, which means that the different nucleotides are being found. While you can use any of the nanopore devices for DNA sequencing, direct RNA sequencing is one of the latest applications. Prep time is cut in half by using the direct RNA sequencing kit sold by Oxford Nanopore Technologies. Some researchers want to remove PCR bias or maybe not use reverse transcriptase. DNA in certain bacteria are copied more efficiently than others, so if you are studying bacterial cell mechanisms, this may be a good technique for you. Stubborn transcripts can be difficult to reverse transcribe, and you can also detect native RNA modifications using this method. This technique can be used on any Oxford Nanopore device, such as the MinION, and is more useful when learning about cellular interactions and epigenetic implications. Module 2 in 611 was all about next generation sequencing. It focused mostly on Illumina, which is pretty good, and second generation sequencing, but Nanopore is considered third generation sequencing. Oxford Nanopore Technologies makes several devices that use this type of sequencing, ranging from thin and portable, as the MinION you can see there in the middle, to slightly bulkier and larger for more large-scale applications. My favorite is the Flongle, just because of the name, but the Flongle is an adapter for the MinION sequencer, which is the portable option. The MinION will set you back about $1,000 for just the device, which isn't too bad, and if you somehow win the lottery, you can get super bougie and purchase the PromethION for the low price of $225,000. In class, we focused on Illumina, and it is considered short-read technology. Nanopore sequencing is long-read to ultra-long-read. You can get all the way up to 26,000 BP reads. Library prep is also simpler than Illumina, and Oxford Technologies offers various kits to make it even easier to prep on the go. There are a lot of advantages to nanopore sequencing over other methods, but I'll just cover a few of them here. Illumina, which again we've discussed mostly in class, is short read, and this has to be done in a lab environment to achieve high throughput. PacBio is another long read technology like Nanopore. However, PacBio and the other long reads need to convert mRNA into cDNA, whereas Nanopore can directly sequence native DNA or RNA. The MinION device is handheld and portable, as well as USB rechargeable, which allows for real-time reads when you just really need to know what kind of RNA you have. Oxford makes another device called the Voitrax, and this is something that they're working on, which gives you automated sample prep. This means you can sequence samples anywhere with fewer lab skills or devices needed compared to traditional library prep, which requires many other steps. Nanopore technology can also process extremely long reads. The MinION specifically has been able to process a read greater than 4 MB in length, although 1.2 MB max is recommended. Other compact devices such as the PromethION, this is not a compact device, that was a lie, sorry, can handle ultra large reads, but the PromethION is the one that you would find in your lab space. You wouldn't want to take that one with you. It costs a lot of money too. Finally, nanopores allow for direct electronic analysis of your DNA or RNA and avoids bias when dealing with the traditional amplification of nucleic acids in regular sequencing methods. This also opens up opportunities for epigenetic analysis, which I will talk about in another slide. The graphic on the left is taken from the Applications and Potentials of Nanopore Sequencing manuscript, and it describes the workflow for detecting methylation sites within a given sample. As epigenetics becomes more prevalent and we learn how these changes can affect organisms throughout their lives, nanopore technology and direct RNA sequencing will undoubtedly become more widely used in research. Viral transcriptomes are usually complicated when sequenced by traditional methods. Direct RNA sequencing does not require recoding and amplification biases that we see with other sequencing methods. In a study conducted in Nature Communications, a research team used direct RNA-seq to profile the herpes simplex virus 1 transcriptome during infection of primary cells. This type of data is extremely important if we want to further understand how viruses can cause disease. In the graphic to the right, we see that the nanopore reads represent a single directly sequenced polyadentylated RNA, compared to the Illumina reads that are highly fragmented. Despite sounding like futuristic technology that solves all of our problems, there are still some challenges and limitations present with nanopore devices, the largest issue being the base call accuracy. More than 100 base modifications of RNA have been discovered, and that is a huge challenge to detect all modified bases. There could also be more modifications that we have not discovered, leading to even more inaccuracies. Some modifications may make nanopore sequencing impossible, such as glycosylation. Accuracy and sensitivity of SNPs detected with nanopore have been higher than Illumina. Tandem repeats and repetitive elements are still being detected, which poses yet another challenge. Currently, direct RNA-seq cannot multiplex, which gives cDNA PCR sequencing an advantage, but this is in development. The throughput is also lower than cDNA sequencing. Finally, the per base cost of nanopore tech is still several times higher than other short read sequencing techniques such as PacBio, and depending on the device you want, you still may need to sell a kidney. Some of the controls that the nanopore device include the run until mode, and this tells the nanopore devices to run and continue sequencing until a certain point has been seen several times at a specific confidence level. So this can be anything you want. For example, if the device can process the sample until a minimum of threefold read coverage over the specific regions you want within, say an RNA sample, or until a specific mutation has been observed a certain amount of times. This way, the user controls the experiment and not the device. The read until mode allows the device to sequence the first bases of a DNA molecule that pass through the nanopore and analyze them in real time. If the sequence is determined to be of interest based on what you have put it in to look for, the sequencing continues. If not, then the nucleic acid will be rejected from the pore and another sequence will be read. Researchers outside of Oxford Technologies have developed multiple quality control methods using programming and computational approaches. Two such instances of these are the NanoR, which is implemented in R, a programming language used by biologists. Another is PyPor, which is implemented in Python, and that is another type of programming language. Based on this information, I will take Dr. Borley's advice and look more into learning bioinformatics languages because that's about all I can tell you related to these two types of things without my brain imploding. The figure on the right shows the workflow for the QC methods NanoR and PyPor that were developed by David Bolognini and team. You start with FASTQ and FAST5 sequencing files that are recorded from the Nanopore devices, run the sequences, and compare them using a program called NanoCompare. Another program called SeqStats can perform data conversions and quality assessments all at once. In conclusion, direct RNA-seq is an important technique that can allow for whole transcriptome sequencing. High-output sequencing of whole transcriptomes can be completed from low input amounts and give you full-length transcripts at the isoform level. This type of technology is extremely useful in studying the cellular mechanisms of pathogens, cancer, and the effects of epigenetic modifications like methylation on genes. If you want to look at what the cell is currently doing right now, consider direct RNA sequencing. Thank you for watching my local expert presentation. Here are my references, and this presentation has been sponsored by Beans, who has been fundamental in helping me learn about advanced microbiological research techniques.